Hi, I'm Tristan Bolts, a third-year aerospace engineering major, and I researched a system that dealt with bioengineering in space exploration because in order to sustain the health and well-being of crews being sent to space to improve and continue space exploration projects, more developments need to be made to the life support systems that are currently in production as well as innovating new bioengineered systems. One of the main facilities for bioengineering in space exploration is Space Biosciences at NASA Ames. It was founded in the early 1960s, and its purpose is to study how the environment of space affects living organisms, engineer ground and flight research facilities in space, develop technologies to sustain life in space, and examine how bioengineered systems can aid human spaceflight. Under the Space Biosciences Division at NASA Ames are three branches: Biosciences, Research, Flight Systems, Implementation, and Bioengineering. In this video, we will be exploring a system developed under the Bioengineering branch. The goal of the Bioengineering branch is to develop innovative technologies for human life support in space. The branch has many different research and technology development areas, including atmosphere revitalization, trace containment control, water recovery, waste management, engineered nanoscaled self-assembling enzyme complexes, systems engineering tools for technology gap identification, in situ resource utilization, as well as many others. One of the projects currently being tested by Mission SpaceX 14 on the International Space Station is a multi-use variable gravity platform validation. It is a hardware platform that is owned and developed by TechShot to address the problem of needing more gravity in space to reduce bone loss and reduce the effects of little gravity has on astronauts' spatial orientation, head and hand-eye coordination, balance, and locomotion. The system addresses this problem by generating artificial gravity from 0.1 to 2 Gs. The system does this by using two carousels that spin quickly to simulate up to 2 times the force of gravity. This is the largest centrifuge in the US segment of the space station that allows researchers to have more control over their testing. The components of the bioengineered system that allow for more control will be detailed later in this presentation. There are three biological aspects of the system that I found to be the most unique. The multi-use variable gravity platform validation permits active environmental control, which includes the temperature and the relative humidity. This feature leads to researchers being able to plan longer and more complex microgravity experiments. Being able to have control over the environment within the system is a crucial part for the development of the organisms being tested. The system is designed for research on many different organisms and cell types, but is currently only being used for Drosophila melanogaster, more commonly known as fruit flies. Because of the versatility of the system to be used for different organisms, it leads to the expansion of the range of life science hardware that is available for space biology research. Cell culturing is an important biological aspect of the system because it offers many possibilities that are difficult to do in vivo. Conditions and parameters can easily be changed and controlled so that valuable data for different scenarios can be analyzed. The size of the multi-use variable gravity platform is close to the size of a microwave and is one of the most important design choices. Space exploration is constantly looking for smaller and lighter designs that can easily be transported into space. Also, area is limited on the space station, so large devices are not ideal because it takes up more room that could be used for other experiments. By choosing to use two independent self-balancing centrifuges in the system, it is able to prevent micro-G loading of specimens as fluids or specimen shift or fluids are added to or removed. Control over the environment within the system makes it more useful because it can be used for different organisms. Other design choices are controlled air convection in each specimen holder, 2 levels of containment, and 12 independent specimen lighting systems are all design choices that aid to make the experiment conducted more controllable and produce clear data. The societal impacts of the platform mainly affect the crew of people sent to space and their families. Astronauts are currently at high risk of age-related fractures decades after their missions are completed. Because of this, they are having to go through many hip and other joint replacements, are returning with the bone strength of older women with osteoporosis, If a large-scale models could be produced for humans use, astronauts could reduce the amount of bone loss in space and could therefore return from their missions with a healthier body and can go on living life without worries of fractures. The fruit fly lab that is currently being tested in the system aids the ecological and societal impacts because approximately 77% of human disease genes have analogs in the fruit fly genome. Thus, the effects of the space environment on the branch of the immune system responsible for quick, non-specific responses to infection can be analyzed and increase human health. The goal is to increase the overall health and well-being of people on mission to space by examining the effects of space environment on innate immunity. Future improvements of the multi-use variable gravity platform can help scientists improve our understanding of biological processes that affect human health on Earth and in space. Currently, more modules are being produced for testing of other model organisms, such as culture cells, Arabidopsis, a family of flowering plants, and aquatics. Current upgrades that are available are specimen mass measurement and air and water filtration. In the future, the technology hopefully can be used for testing in missions beyond low Earth orbit and will be of a larger size. Here is a list of the references that were used in this presentation, and thank you for watching.